Hi, my name is Mochi. I am a first-year graduate student in the quantitative computational biology program at Princeton University. I will be talking to you today about behavior trigger analysis in the nematode C. elegans. I am interested in how animals make behavioral decisions given sensory information. We're working with the model organism C. elegans, which is a great organism to study the neurosensory transformations that underlie the basis of behavior, because one, it responds to a variety of sensory stimuli, such as touch, temperature, and chemical cues. Two, it exhibits a diverse but manageable sets of behaviors, such as reversals, pauses, and egg laying. Three, it is genetically tractable. Four, it has a relatively simple nervous system. In fact, it is the only animal with the connections of its entire neurosystem map. And five, it is optically transparent, which enables us to use all sorts of optical tools. One of these interesting behaviors is the mechanosensory response, escape response. In this video, we see the worm being touched by an eyelash, and it performs a reversal followed by a change in the direction it's going. I chose to study it. The mechanosensory escape response is very important behavior to the worm because it allows the worm to escape predation. I chose to study the mechanosensory escape response because it is a fairly representative behavior. Many organisms exhibit it. It is well studied in literature, and it's stereotyped and easy to detect. All I need to do is look for a change in the direction of the worm. Even though I will focus on the mechanosensory escape response in the following experiments, the techniques I described can be applied to any stereotype behavior. I am not the first one to study the mechanosensory escape response. Previous studies have focused on individual neurons through ablation and mutation studies, which provides us with a circuit diagram. The key sensory, inner, and motor neurons involved are shown here, but the list is not exhaustive. The final output of the network are behaviors such as reversals we saw in the previous slide. These studies have taught us a lot about the neurocircuitry. For example, we know that the mechanosensory neurons AVA and ALM express a pressure-sensitive ion channel that allows the animal to sense touch. And that the inner neurons, including AVA and RIA, process information from sensory neurons to coordinate the motor output. However, there are severe limitations to studying the sensory motor transformation relying on these fundamentally static techniques, such as behavioral mutants and laser ablations. For example, in laser ablation experiments, a single neuron is destroyed by the precise pulse of laser. The worm is then observed for behavioral defects, but the original neurocircuitry is fundamentally altered and permanently defective. Experiments like these tend to miss the interesting neurodynamics, like how the network computes differently based on its changing internal state. What I want to do is I want to take a step back to the bigger picture. Forget the names of the neurons and model the entire decision-making neurocircuitry using a relatively simple model and see how the parameters of this model changes with the network state. To do this, I am breaking this into three modules: the stimulus input to the sensory neurons, a behavioral output in the form of reversals, and a black box containing all the inner neurons and motor neurons as a single computational unit. So similar to what Matt has done in Drosophila olfactory, we're given some sensory input. I would like to predict reversal decisions from the escape response using a simple model. And let's take a look with how we're going to model this black box. We'll employ a linear nonlinear Poisson cascade, which has been used extensively in neuroscience to model spiking neurons. And it has two fitted parameters, a linear kernel and a nonlinear function. to make reversal predictions using the model, the sensory input stimulus is first filtered by convolving with this linear kernel, and the resulting filter signal is converted into a reversal rate using a nonlinear function. In other words, at each time point, there's a changing probability of reversal based on the model parameters and previous stimulus input. I will talk about how the parameters are fitted in more detail later on in the presentation. But first, in order to fit the parameters, I will need to be able to control the sensory input with precision and collect a lot of data, neither of which is possible using an eyelash, which is why I need to introduce optogenetics. Optogenetics uses engineered channel proteins that are light sensitive to control neuractivity. In our case, we genetically engineer worms to express the optogenetic protein crimson only in the mechanosensory neurons. When exposed to red light, crimson opens and allows sodium to flow into the cell as if the pressure-sensitive membrane channels have opened. This means that the worm will feel like it's being touched despite the fact that it's not being perturbed physically. Here's a video of the mechanosensory escape response evoked optogenetically. This came from the work done by my PI, Andrew Leifer, in the earlier study. As you can see, the worm produces the same type of behavior as if it was touched by an eyelash. So in order to fit the parameters of the NIN LMP model, which requires a lot of data, we have constructed this apparatus to help with the daily collection by imaging and tracking 30 to 50 worms in parallel doing a single experiment. The custom-built apparatus simultaneously images and stimulates at 14 frames per second. The stimulus is delivered by a single red LED to the entire plate. The plate is illuminated by an infrared ring light, and the illumination changes due to the stimulus is filtered out by an infrared long-pass filter. So here we have some example data. Around 30 transgenic worms expressing the optogenetic protein crimson is put onto a 10-centimeter plate. Each dot is a worm tracked in time. The stimulus here is a simple 2-second pulse of the intense red light, and which is on when the red dot appears in the upper right corner. The tracking algorithm identifies the reversing worms and plots them in red. Now that we have introduced all the tools we need to fit the parameters, I want to go back to the LMP model and talk more about how we fit these parameters. The first parameter, the linear kernel, can be found by reverse correlation. That is, whenever one decides to reverse, we look at the sensory input immediately prior to the reversal and tend to correlate the behavioral decision back to the sensory input. Let's take a look at how this works in action. The image on the left shows the actual behavior of the worm. The top graphs shows the speed and the angular speed of the worm, which are used to identify reversals. The reversal bouts are shown in these red boxes. The third graph, this graph, shows the stimulus delivered to the animal. Instead of a simple pulse of light as seen before, the stimulus now is a Gaussian white noise. This is because if the input is a Gaussian white noise, the math works out so that the linear filter that we're fitting is simply the time reversal triggered average. In other words, whenever we see a worm reverse, the analysis code will take the Gaussian white noise and average it and align it in time so that the reversal occurs at time 0. Hence, reversal trigger averaging. This is shown in this graph. Whenever the worm reverses, the stimulus from the third graph gets averaged into here. And as you can see from this demo, the reversal triggered average is initially extremely noisy due to the nature of the stimulus, but as more events gets averaged in, we see a characteristic shape of the linear filter beginning to form. Here is the stimulus intensity averaged over thousands of events, aligned so that the reversal occurs at time 0. On the left, the horizontal line indicates the mean of the stimulus intensity. As you can see in the left graph, there's no correlation between the stimulus and the behavior in the native control, which have disabled optogenetic protein channels. In other words, the worm's reversals are not correlated with light stimulation. In the experimental condition, however, the reversal triggered average and thus the linear kernel looks strikingly similar to the shape of a Gaussian derivative operator, which suggests that the animal is constantly looking for positive changes in light intensity to decide when to reverse. Because the stimulus and behavior appears uncorrelated beyond 6 seconds before the reversal, the linear filter is only 6 seconds long, which also informs us about the time scale the animal is keeping track of the sensory information. After fitting the linear filter, we'll need to find the second parameter, the nonlinear function, which converts the filter signal into a probability at each time point. This is done by simply taking the histogram of whenever there's a histogram of the filter signal whenever there is a reversal, and dividing it by the histogram of all the filter signals. We can then fit an exponential or sigmoidal function to the data. Here is the nonlinear function for our reversal triggered analysis experiment fitted to an exponential function. On the x-axis, we have the filter signal in arbitrary units, and on the y-axis, we have a predicted reversal probability. Using this function, we can map a filter signal to a predicted reversal probability for each time point. So after fitting the parameters, we want to ask if the LNPs can actually predict reversals, just as a sanity check. To do this, we will perform a cross-validation. We randomly take half the data from the LNP to fit the LNP parameters and see how well it predicts the reversals in the other half of the data, and give it a score. The score is based on how well the LNP predicted reversal rate matches observed reversals. A higher score indicates better predictions. The shuffle score is generated by randomly shuffling the observed reversals before calculating the score, generating a null model that the LNP should not predict. Repeat it 1,000 times, and we created this histogram. And we see that the LNP model predicts reversals significantly better than the null. So after seeing that we can predict behavior with the LNP model, the next question may be, how might this be useful? Allow me to give you an example by quantifying habituation using the first LNP parameter, the linear kernel. On the left, we have the reversal triggered average for the first 10 minutes of the Gaussian white noise experiment. And on the right, we have the reversal triggered average for minutes 20 to 30. As you can see, we can measure the desensitization to the stimulus by simply looking at the decrease in the positive load of the fitted linear kernel. So in conclusion, the behavior triggered analysis provides a powerful tool to describe decision-making based on sensory input. It can also be used to quantitatively measure changes in neural computation depending on the worm's neurodynamical state, as exemplified in the slide before, providing us with clues to the underlying mechanism. And with behavior-triggered analysis, I hope to study topics such as habituation, multisensory integration, and learning in the future. So with that, I want to thank you all for listening to my talk, and my advisors, Andy and Josh, along with other members in our labs, especially George and Jose for helping me run the experiments, and my collaborator, Mark Kershaw and NYU for his input, and I appreciate any feedback and questions from you guys. Thank you.
Hi, thanks for the talk. So you had this plot that said that you're predicting better than the null.
Right.
How well do you think you're predicting, or put differently, how better is it, would it conceivably possible to predict than how well you're doing with your analysis?
So
are you getting it every time?
So, so to answer that question, I'm not sure if the LNP model is the best model to predict behavior. So I'm looking for other models to compare the LNP models against, so that we can have an idea of how well it's actually doing against other models.
Right, so like if 0 is the null and 100 is perfect, where are you guys where you are?
So I don't know if you could do that, because what the LNP model essentially is generating is a probability, and what you actually observe is discrete events.
So, very nice application of LNP models. I'm curious, though, in your black box and the dynamics that you're using, you are having included in that the transformation of your optical signal into channel activity through fencing, which is different than mechanical sensation in the natural words behavior. So you sort of, in your linear filter, you included the dynamics of the optogenetic channel. So I'm curious if you think of those like so fast or so linear that you're not concerned that they're going to influence the shape of your filter compared to what the filter might be if you gave a mechanical white noise stimulus and used the natural mechanoreception.
So again, I am not sure what that linear filter would look like if I actually gave it a white noise stimulus using eyelash. That is a hard experiment to do. So really, it's not comparable.